Hi, folks. My name is Ben Carter, and I'm a professor up at San Jose State University, and I'm a specialist in bryophytes, and so that's what I'm going to be talking to you about today. And the outline of the lecture will go over the evolutionary origins of the three bryophyte lineages, and then talk about the differences between bryophytes and other plants. Then we'll talk a little bit about the life cycle of bryophytes and some of their basic structures. And then we'll get a little bit into the ecophysiology and talk about why bryophytes are not just little plants ecophysiologically. So to start off with, a lot of people, when they hear that I study bryophytes, they ask sort of what they're good for. And other than just being interesting organisms in their own right, they do have some pretty critical ecosystem roles around the world. So for example, at high latitudes, where you get the large expanses of peat bogs, bryophytes and both living and dead tissues actually form the largest carbon sink in the world, so more even than in the Amazon rainforest and places like that. In the deserts, on the other hand, you get really tiny bryophytes that form these desert crusts, and these are really important for preventing erosion as well as preventing the spread of invasive species. This is something that a lot of people are doing research on here in the southwestern U.S. And then especially in cloud forests and tropical montane areas, you get big bushy bryophytes that have a really important role in the water cycling and the nutrient cycling. So you can basically get this in places that have branches draped with bryophytes, and so you can, after a rain event, you can store hundreds of thousands of liters per acre of water in the bryophyte tissues, and so they kind of absorb it all, and then they slowly release it, and so it totally changes the humidity and nutrient cycling of the whole ecosystem. And then finally, everywhere in the world, just like the bigger plants that you're used to looking at, provide really important habitat for all different kinds of animals and fungi. Bryophytes do the same thing at a smaller scale. So everything from mites that you can see with the naked eye, all the way down to microscopic organisms like water bears and rotifers and a whole other host of other organisms basically rely on the kind of peculiarities of bryophyte physiology and the way they wet up and dry down, which we'll talk about for their homes. So I want to just kind of start out with looking at this evolutionary tree, and so I'll show this to you in a number of forms over the next few slides. And so you can see down here is the ancestor of all plants that was probably something sort of similar to the green algae today. And then branching off first are the three bryophyte lineages, and we'll talk more in detail about these in just a slide or two. And then we up here, so after maybe 450 million years ago, roughly, which is when this was happening, you get into probably the plants that you're maybe a little more familiar with. So somewhere around in here was the origin of vascular tissue, or at least one of the origins of vascular tissue, which we'll talk about, and also physiologically significant stomata. So bryophytes actually do have stomata, but it's on the sporophyte generation, so they're not really important. And then you also get the evolution of roots and of sporophyte dominance. So in the alternation of generations, there's a haploid generation and a diploid generation, and so this is where the haploid generation becomes dominant. And then somewhere along this branch, you get the origin of leaves with branched venation. So this is what separates the lycophytes, which have small scale-like leaves, from the ferns, which have much bigger leaves. And so you can imagine, if you look at leaves of flowering plants today, how important the evolution of branched venation was. And then on this branch here, we get the origin of seeds. So that's what separates the angiosperms and gymnosperms from the ferns and lycophytes. And then within the angiosperms, which are the very large majority of all living plants today, you get the origin of flowers and fruits. And then they're not unique features, but other important features of the angiosperms are the presence of vessel elements, which are really big structures within the xylem, which allow much more efficient water transport. And then also a whole host of mycorrhizal associations, so relationships with fungi that are either absent or much less well developed in all these other plant lineages. And so some combination of flowers and fruit and vessel elements and mycorrhizae probably leads to the massive diversification of the flowering plants that we see today. But today, we're here to talk about the liverworts and hornworts and mosses. And so you can see from this phylogeny that there are 1, 2, 3, and then 4 major lineages of land plants. And so bryophytes, in terms of evolutionary history, actually comprise 3 out of the 4 lineages of land plants. So what separates land plants from the algae, both the green and red algae, which are close relatives of land plants, is the presence of an embryo. And so the technical name of the land plants are embryophytes, or plants that have embryos. And so the embryo, just like in an animal, is the situation where you have a zygote, so you get the egg and the sperm fusing together to form a zygote. And so if that zygote is maintained inside of maternal tissue, we call it an embryo. So this is in contrast to organisms that disperse their eggs into the water. So some algae do this. Some animals like fish will do this. So spawning fish like salmon just put their eggs and their sperm right out into the water. But so you can imagine how the development of an embryo would be important in the kind of the conquest of land, when these plants first kind of came up out of the water and became terrestrial organisms. So here are the 4 main branches on the tree of life as they exist today. There's some other extinct branches in there. So the 4, and you can notice there are these dotted lines down here that I'll talk about in just a minute. But we've got the liverworts, the hornworts, and the mosses. So liverworts, hornworts, and mosses in no particular order. And then this other group is what bryologists refer to as the atoc clade. And this is just an acronym for all that other crap, which is sort of comprising the rest of plant diversity in our view. And so about these dotted lines down here, these are meant to reflect accurately that we really don't know what the particular relationship is among these 4 major lineages. So it could have been that liverworts diverged first, or maybe that hornworts came off first, or maybe mosses actually are sister to liverworts, or maybe hornworts are actually sister to vascular plants. We just don't really know. And the reason for that, it's not that we haven't tried, but it's that this event or series of events happened a very long time ago, so somewhere in the neighborhood of 450 million years. And also probably happened relatively quickly. And so there just wasn't a lot of opportunity for either anatomical evolution or just nucleotide substitutions or mutations to arise between when these different lineages split off from one another. And so there's just not really good support at the base of the evolutionary history of plants. And so it's quite possible that we may never know. And basically there are published studies that basically give you all possible combinations of these 4 lineages in terms of who's sister to whom and which one diverged first and second, etc. So it's unresolved and unlikely to be resolved, even with the biggest genomic data sets that we have. So then traditionally, bryophytes have been separated from vascular plants. So if you take these 3 lineages and separate them from these lineages, you can see here it says non-vascular plants. So that's been kind of classically the trait that we use, but it's a bit of a misnomer because although it's true that vascular plants have vascular tissue, some of the non-vascular plants actually have functional vascular tissue as well. And it appears to have evolved independently, but we're not entirely sure of that. But either way, it's not really a good synapomorphy. So the more kind of proper synapomorphy that we've come to recognize recently is the presence of a branched sporophyte. And so the sporophyte generation, again, we'll talk about the life cycle in a minute. But so when you look at a plant, it's usually somewhat branched. So it has a branched sporophyte, where in bryophytes, the sporophyte is always unbranched. And so again, if you look out your window and look at some trees or other plants growing around, you could imagine how important a branched body would be. So that certainly was a key feature and a really good anatomical feature that actually separates out the bryophytes from the what we call quote unquote vascular plants. Here's just an image of the largest mosses in the world. And so you can see they're about knee high. These are species that occur in Papua New Guinea and New Zealand and places like that in the southern hemisphere. And they actually have fully functional, we don't actually call it xylem and phloem, but if I showed you a cross-section of its stems, you could easily identify the cells that look very similar to what we know of as xylem and phloem. And so it's apparently an independent evolution of those tissues. And it also says something about the limitations of water transport in plants. And so if you give mosses vascular tissue, they get big, just like a quote-unquote vascular plant would. So in general, even though the bryophytes comprise these three different lineages, we tend to think of them sort of ecologically and sometimes even evolutionarily as a group. And that's because they maintain a number of not synapomorphies, but shared characteristics that just relate to their ancient ancestry. So some of these characteristics are, and so these are shared by all the bryophytes, and they're different from certainly from flowering plants, and in some cases even from all of the other quote-unquote vascular plants. But the differences are that bryophytes have a dominant gametophyte or haploid generation, and again we'll talk about life cycles in a minute. They're dispersed by spores, so unicellular spores, not seeds. So this is something they actually share with the ferns and the lycophytes. They do not have flowers or fruit, again similar to ferns. They do not have roots, so this is a difference between bryophytes and ferns and lycophytes. So the latter two do have roots. Stomata only on the sporophyte generation, and so ecologically unimportant. So all the other plants have stomata on the sporophyte generation as well, but in the case of all the other plant lineages, those sporophytes are dominant. So they're the kind of ecologically important life stage. And so since bryophytes only have stomata on the ecologically unimportant life stage, the stomata really aren't very important, which is quite different from the rest of the plant lineages. Sperms swim through the environment to get from male to female gametangia. So basically, again, the sperm get broadcast into water, and then the sperm actually swim through a film of water to the female gametangia. And so this is one of the reasons, not the only reason, but one of the reasons why you tend to see mosses in wet places is because they need water, liquid water for fertilization. And again, this is something that ferns also do and lycophytes, but the evolution of a seed sort of makes that unimportant for the seed plants. Another number 7 down here, similarity among the bryophytes is that most of them lack specialized vascular tissue. So again, there are a couple of lineages scattered throughout, but it's ecologically not an important trait. And then most of them, or at least a lot of them, are what's called desiccation tolerant. And we'll talk about that at the end of the lecture, but that basically means they can dry out and kind of revive themselves. Unlike most vascular plants where when you dry them out, then they're just dead. So we're going to shift gears into the life cycle. So all plants undergo something called alternation of generations. So you basically have a haploid, so that has a single complement of the genome. So haploid gametophyte, that means a gamete-producing plant. And that is the parent of a diploid, or 2N, sporophyte, or spore-producing plant. And then those spores produce haploids, which then produce gametes that produce diploids. And then those diploids produce spores, which produce haploids. And so you get this alternation between haploid and diploid and haploid and diploid, sporophyte, gametophyte generation. And seed plants, at least the gymnosperms, still technically have this difference, but it's almost evolved out of existence in the angiosperms. So if you didn't know where to look for it, for example, in pollen tubes and inside of the developing ovule, you'd have a hard time knowing that flowering plants came from ancestors with alternation of generations, but there are some telltale signs. In bryophytes, the differences are very obvious. So the gametophyte is the leafy green structure, and then the sporophytes are these little structures up on stalks. So this is probably one large gametophyte, sort of analogous to like a big bush, except it's haploid, not diploid. And then each one of these stalked things, each sporophyte is a separate diploid individual that is actually parasitic on its gametophytic mother. And so this is pretty similar to the life cycle of ferns, where you get this is about a thumbnail sized flap of tissue that's the gametophyte generation. So this is haploid. And a sperm will swim from another one of these structures into this one, fertilize it, and so after that fertilization event, you get the development of the sporophyte generation. And so you can see this looks kind of like a little tiny baby fern frond, and that's exactly what it is. And so a big difference between bryophytes and ferns is that this one quickly will put down roots, the sporophyte generation, and grow up to be very often a long-lived perennial diploid individual, whereas these gametophytes just persist for a couple of weeks in the environment. And the mosses, on the other hand, have usually a long-lived gametophyte generation with a pretty wimpy little sporophyte that's basically just there for a season to produce spores. So here, as promised, is a life cycle. So we'll just go through this really quickly, but it's very useful to understand the difference between bryophytes and the vascular plants. So you can see this dotted line that goes through the middle of the diagram. This represents the separation between the haploid or gametophyte generation and up on top, and then the diploid or sporophyte generation on the bottom. And then there's 2 points right here and here where the life cycle transitions between haploid and diploid. So when you go from a diploid to a haploid, you undergo meiosis. So it's a splitting of a diploid cell into multiple haploid cells. And then on the other side, you have fertilization, which it comes from the fusion of 2 haploid gametes to form a diploid zygote. And so all life cycles have these basic 4 components: haploid, diploid, meiosis, and fertilization, but they're very different. So for example, in people, we're diploid, and then we undergo meiosis to produce gametes. So we basically skip this whole stage and go from the gametes straight to fertilization. So we basically completely gotten rid of the haploid generation, and that's very similar to what flowering plants have done as well. So in general, the way this works is you start with haploid spores. Those go out into the environment, fall onto the soil, and produce what looks like a filamentous green alga, but it's actually just a mass of what we call protonema, which are green filaments, so green kind of linear cells that make this sort of mesh or web. And then out of that will grow gametophores, and so these are the leafy branches that are recognizable as a moss or liverwort. And so these can be either unisexual or bisexual. And then on these leafy structures, you get the production of sperm, as well as the production of eggs. And then this is basically a sac of sperm, and that will rupture when the time is right. And that will emit the sperm into the environment, where they'll swim, assuming there's some water around. So they'll swim through, and this can just be like on a rainy day. So they'll swim through the water down this canal to where the egg is in the female structure. And so that's where you get fertilization and the formation of a zygote. And then once that zygote starts undergoing cellular division, it's an embryo. And then that developing diploid structure. So again, the mother is haploid and the offspring is diploid. And that will grow up into a stalk in the case of a moss, and become the sporophyte. And eventually the sporophyte will undergo meiosis and produce from diploid progenitor cells. It will produce haploid spores again, and then you make it all the way around. And so this structure lives in the environment independently, and then this sporophyte is actually parasitic on its maternal gametophyte. So that's the kind of the basic life cycle without too much detail. So these are all the kind of the things that hold bryophytes together as a group. And so now we're going to kind of focus on what the differences are between the three lineages of bryophytes. So the names of the three are the mosses, the liverworts, and the hornworts. The global numbers, so the mosses are most diverse with about 12,000 species, and here in California we've got about 650. Liverworts have about half that number, so about 5,000 species globally, and much fewer in California, about 150. And then hornworts have about 100 species globally and only around 6 in California. So if you add these up, in California we have about 800 native bryophyte species. And if you compare that to the vascular plants, this actually comprises about 15% of California native plant species. So it's not a trivial number. It's actually a pretty substantial number in terms of the overall plant richness in California. So now I'm just going to go over a couple of slides each for each of the major lineages, the hornworts, the liverworts, and the mosses, and show you some of the basic anatomical and morphological differences in both the gametophyte and sporophyte generation. And we'll talk more about this in lab. So we'll start with the hornworts. So the gametophyte generation, so that's this kind of shiny, it's wet in this image, but this kind of shiny, a lot of people refer to it as looking sort of like a melted gummy bear. It's kind of translucent like that. So that's the gametophyte, and then these little spears that look like baby grass, those are the sporophytes. So the gametophyte has an undifferentiated thallus, so it's just basically these kind of like straps of tissue that are not differentiated into organs. They have either 1 or 2 chloroplasts per cell, and this is an important difference between the hornworts and the thalloid liverworts, which also have a thallus but have many cells or chloroplasts per cell. Hornworts all have a nostoc, which is a cyanobacteria, as an endosymbiont, so it actually lives inside the cells, and that's one of the things that gives it that kind of bluish green translucent look, which is quite different from thalloid liverworts. The sporophyte, again, is like a needle-like structure, and it opens by 2 longitudinal slits that run up the sides of the sporophyte. So here's an image through a microscope of what hornwort cells look like. So you can see they have this really big single, or every once in a while they'll be double chloroplasts. So instead of having a whole bunch of little chloroplasts, they have one really gigantic one, or sometimes 2, right in the middle. So they're very easy to tell under the microscope. This is what they look like when the spores are being produced. So here's the kind of immature sporophytes, and then as they mature, you can see these kind of paired, it looks like dead grass. Those are the 2 valves that split apart. And then you can see that it starts maturing at the tip, and then it matures down toward the base. And so this whole structure will mature and then emit spores until it gets all the way to the base down there. So pretty distinctive. And you can get a sense, this is a typical sized one, so this is in the palm of somebody's hand. The second major group is the liverworts. So the gametophyte of liverworts can actually be thalloid, similar to the hornwort, where you just get straps of either completely undifferentiated or only lightly differentiated tissues. Or in some other liverworts, you can actually have a stem and leaf architecture, which is similar to a moss. So In liverworts, there's just been a lot of morphological evolution, and so a lot of evolution in the body plan. And again, we'll talk about this more in lab of all the different kinds. But so a liverwort you could potentially confuse with either a hornwort if it's thalloid, or a moss if it's got stem and leaf morphology. But some of the other characters you can use is that under the microscope they have a unique organelle called oil bodies, and we're not entirely sure what these are for, but they're there in most liverworts. Now I'll show you an image. So they're absent in mosses and hornworts and all other plants and all other living things. Unlike the hornworts, liverworts have many chloroplasts per cell. So if you get a thalloid structure, you can just throw it under a microscope and see if it's got 1 or 2 chloroplasts or many chloroplasts. And then for the stem and leaf morphology that you might confuse with a moss, the leafy ones typically have 2 to 3 longitudinal ranks or rows of leaves going down the stem, and so they're not spiraling like in a moss. So moss are arranged just like in regular plants, where in most cases the leaves kind of spiral down the stem. And then the sporophyte is typically either spherical or egg-shaped, and it splits open by 4 valves, and often it's stalked. So here's some images of typical liverwort sporophytes. So you can see this kind of transparent stalk, and then each one of these little kind of olive-like structures, these little balls, that's the capsule. So that's the sporophyte. And then when it opens, it just splits apart into 4 different pieces, and those emit the spores. Some liverworts, and these are often the ones that you see in pictures, look like this with this kind of umbrella-like structure called an archegoniophore. Here's another version on a different species of these little kind of umbrella-type structures. And in these cases, that umbrella is actually gametophyte tissue, and then embedded within this little white cone. So if you were to turn this over and look up inside there would be one of those little black olive-like capsules. So you can see, like, similar to these naked capsules. Those are going to be embedded in that gametophyte tissue. So they still have the black balls, but they're just kind of tucked into the gametophyte tissue. And here's some images of what liverwort oil bodies look like. And again, these are those unique organelles that no other organisms have. And so these are images of cells taken through just a light microscope. So you can see each one of these cells has chloroplasts squished up around the edges from the vascule kind of pushing them to the walls. And then in the middle of the cell, you can see these big oil bodies. And so here's some blue ones, and these ones are very granular. And so basically the number and the shape and the texture are all really important for taxonomy of liverworts. And again, we're not entirely sure what the function is, and it's one of those things it's kind of hard to know the function, because pretty much all liverworts have them and nothing else has them. So there hasn't really been any kind of evolutionary replication that we could use to test hypotheses about why they originated. But the hypotheses generally kind of float around anti-herbivory, but we're not really sure whether that's true or not. Another really important feature of all bryophytes, really, but liverworts in particular, is a lot of them make asexual propagules. And so that's what these are. If you've ever seen, this is a common weed around California. And each one of these little bud-like structures, this is not a spore or a seed for that matter. This is a little piece of gametophyte tissue that's just budded off, and the purpose is to just get hit by a splash of water, or kicked by somebody walking by, and then each one of these little bundles of tissue could potentially make a new colony. So here's some images of a leafy liverwort. So this has the stem and leaf architecture that you might confuse with a moss. And the difference is that in liverworts, like if you look at one side of a leaf, or sort of a leafy stem, and then turn it over and look at the other side, they're quite different. So here's like the top-down view. So you get 2 ranks or rows of big leaves, and then if you take that structure and flip it over, then you see a third row that's quite different from the first two rows going down the other side. So it has 3 ranks or rows of leaves. So 1, 2, and then 3 on the other side going up the stem, which again is very different from what you see in mosses that just have typical leaves that spiral up the stem. And that brings us to mosses, so the third out of the 3 groups. And so in mosses, the gametophyte morphology is first of all separated into 2 general categories, the acrocarps and the pleurocarps. And I'll talk more about that in a minute. But this is sort of analogous to our separation between monocots and eudicots in the vascular or the flowering plants. So kind of a big separation. Mosses all have stem and leaf architecture, so they're not thalloid. Most have spirally arranged leaves. There's a couple of exceptions of plants that have distichous leaves, or sort of like an iris where they come out in a single plane. But for the most part, they're all spiral. And so the acrocarps are mostly unbranched. So it's kind of like these plants down here. You get these little sort of like little bud type arrangements with a tiny stem and just leaves around that stem. Or you can have pleurocarps, which have a prominent main axis and then side branches coming off that, and then sometimes side branches coming off those side branches. And so there's a difference in the gametophyte morphology as well as the sporophyte orientation. So in the cases of acrocarps, typically the sporophyte emerges from the apex of the stem, and then in pleurocarps, it appears that the sporophytes are axillary, so they're coming out from little side branches, but they're actually, technically they are terminal, but they're just terminal on a very short side branch. But anyway, it gives a very different kind of overall look as to the placement of the sporophyte on the gametophyte. And the sporophyte is a stalked capsule. So it's got a capsule, or a stalk rather, and then the capsule is attached to that stalk. So here's the capsule and the stalk. And instead of opening by slits, 2 slits in the hornworts or 4 in the liverworts, it has a circular opening, sort of a pore. And that circular opening typically is surrounded by specialized teeth called the peristome. I'll show you an image of that in a minute. So here's just a few images that show you the kind of morphological diversity of both the gametophytes and the sporophytes. So here's a pleurocarp that has lots of branching. So each one of these things is a leafy stem. And then here's an image of a plant that has these little kind of star-like gametophytes. And then here's the sporophyte coming up out of it. Again, here these kind of curvy leafed gametophytes with the stalked sporophyte coming out of it. And then here's some gametophytes that have a long on each leaf. And this image does not have any sporophytes in it. The spores are dispersed again through a circular opening, and then this circular opening in this case is surrounded by 2 concentric rings of teeth. And so all the teeth together collectively are known as the peristome. And so they're really important for dispersal of the spores, and they're also really useful in moss taxonomy as well. So a lot of our kind of higher level taxonomy of families and orders and things is based on features of the peristome that tend to be evolutionarily conserved. And I'll tell you more about all those structures in the lab when we get a chance to look at some actual organisms. So then I want to just kind of end with a couple notes on the ecology of bryophytes. And so out of all the differences, and there are many differences between the quote-unquote vascular plants and the bryophytes, 2 of them stand out particularly. One is that most bryophytes are what's called poikilohydric and desiccation tolerant. And so I'll define those on the next couple of slides. And then the other feature that we won't talk about too much, but that is certainly important, is just the issue of scale. So bryophytes tend to be very small, and so their interaction with things like wind and evaporation rates and surface tension of water tend to be very, very different from the larger organisms. So think about your typical moss growing on a rock, and so the sort of relative importance, for example, of gravity, which is going to usually pull water down, versus forces like surface tension, which can pull water in any direction, are going to be really different, just the ratios of those physical forces or properties of water. So poikilohydry is an ecophysiological strategy in which an organism's internal water balance equilibrates with the environment. And desiccation tolerance is the ability to withstand periods of extreme dryness. So it's basically the ability to go dormant in response to stress. And so these plants are basically able to, because they can equilibrate to the environment in terms of their water content, they can avoid drought by kind of using an opposite strategy from vascular plants, which is to just completely dry down and go dormant as opposed to getting stressed out. And so this is kind of analogous to ectothermy, and the difference between ectotherms and endotherms, and then also between heterotherms and homeotherms, if you're familiar with those terms from talking about animals, and their heat regulation. But in bryophytes and vascular plants, we're talking about water instead of heat. So in an organism like a mammal, like Mr. Trump here, you've got what's called a homeothermic endotherm. So homeothermic means that they maintain essentially a constant body temperature, and endotherm meaning that that temperatures is created from within. So it's not getting temperature from the sun. It's producing its own heat, as opposed to something like this frog, which is a heterothermic ectotherm. So heterothermic means that its temperature fluctuates a lot, and ectotherm means that it gets its heat from the external environment. And so you have these very different strategies. So things like mammals are able to be active at night, be active during the cold. So there's kind of all these different sort of landscapes where they can persist, where something like an amphibian cannot. But the trade-off is that it costs a lot more energy. So basically, mammals have to take in a lot more calories than a frog. And so the reason that we're talking about all this is because it's kind of analogous to the difference between how vascular plants and how bryophytes deal with water. So in a vascular plant, if the environment dries off, or dries out, then the vascular plant has to expend a lot of energy, a lot of resources in maintaining its internal water balance. And so if we look at the adaptations of plants in a place like Southern California, where it's really dry, most of those adaptations are dealing with the problem of how do you maintain internal water balance when it's really dry outside. And this is a problem that bryophytes don't have to solve, because when it gets dry outside, basically the internal water pressure in a bryophyte just equilibrates with the external water pressure, which is zero. So when it gets dry, the mosses just go dormant, and so then they just avoid that water stress. And so there's trade-offs. They can't live in certain kinds of environments, and they're not good competitors. And so they're more on the stress tolerator end of things. But it's certainly a case that they're not just sort of like scaled down vascular plants. Like physiologically, the way that they interact with water, water availability, and humidity and wind and everything else that we think of kind of plant ecophysiological strategies dealing with, they attack those problems in a very, very different way, which is one of the reasons that has allowed them to persist probably for the last 450 million years and continue to generate new species. And there's a lot of diversification in mosses. So it's not like they're just some ancient lineage that produced a lot of species millions of years ago and then haven't continue to produce new species. Even in arid areas like California, there's some lineages that are actively undergoing speciation today. So just to summarize a few of the kind of main points. So bryophytes are not one, but three distinct lineages that we kind of lump together: the mosses, the liverworts, and the hornworts. And they diverged from the fourth lineage, the vascular plants, a long time ago, about 450 million years. Bryophytes share a dominant haploid generation and sperm that swims through the external water in the environment. And these are two characteristics they've retained since they diverged from one another. Bryophytes have more species than any major plant lineage except for flowering plants. So the number of mosses and liverworts, for example, is much higher than the number of ferns or gymnosperms or lycophytes. So just because they're small doesn't really mean that they're sort of any less evolutionarily interesting. So there's still, again, like I just mentioned, kind of popping off new species all over the place, and dealing with a changing world in very different ways from the ways that vascular plants are dealing with it. And that kind of leads to the final point here, which is that bryophytes are not just small plants. Their physiology is really, really different from vascular plants. So that's what I've got for you today, and hopefully I will see you in lab.